Good morning, candidate. Hereby I open this academic ceremony in which Mr. Agorastos Agorastos, surprising, will defend the academic thesis entitled The Balance Within, Factors Influencing Neurovisceral Autonomic Responsiveness to Endocrine and Pharmacological Stress Challenges. I would like to invite you to present a summary of your study and basically also the conclusions of your thesis in the next 15 minutes. And I'd like to give you the word.
Highly esteemed Pro-Rector, highly esteemed members of the opposition, dear supervisors and dear colleagues, it is my exceptional honor to be standing in front of you today in this wonderful hall of the Maastricht University to defend my thesis entitled The Balance Within. In the next 15 minutes, I will try to give you a brief introduction into this conceptual framework of my experiments, a little glimpse into the methodology, and a brief overview on the findings and implications of my studies. Ladies and gentlemen, the concept of inner balance is not new. It has been introduced over 2,500 years ago by several ancient Greek philosophers like Pythagoras, Hippocrates, and Aristotle, who suggested that it is actually the balance between opposing forces, either in nature or in the organism, that are responsible and necessary for the maintenance of harmony and health. This concept has been revisited several centuries later by modern scientists like Claude Bernard, who coined the term milieu intérieur, and of course, 50, 60 years later, Walter Cannon, who was responsible for the term homeostasis as being the optimal functioning set point within an organism. And of course, he proposed that the first priority in all living organisms is the maintenance of this homeostasis. However, the maintenance of homeostasis is not simple, and it's rather hypothetical and than an actually achievable set point. Because more or less, our whole lives is spent in a state of threatened homeostasis, which is called stress, a term introduced by Hans Seele and borrowed from the physics. We have developed a highly sophisticated system, the human stress system, in order to be able to re-establish this threatened homeostasis by stressors through adaptive responses in our body. And this stress system is the most central and vital homeostatic system in our organism, and its dysregulation, we know today, it's responsible for pretty much the majority of all chronic non-communicable diseases in men, but also associated with the dysregulation of all subordinated homeostatic systems like the cardiovascular, the metabolic, and the immune system. The human stress system shows central and peripheral components, central as in the brain and peripheral in the periphery of our organism, and shows a top-down regulation. This means that the most advantaged parts of our brain influence the function of older ones, and of course the peripheral limbs of the stress system, which are two. The main two limbs are first the one responsible for the first wave of the stress response, and which is initiated by nerves. It's called the autonomic nervous system, with the two branches, the sympathetic and the parasympathetic nervous system, who are pretty much in an interdependent balance to each other, which is very important to every reactive procedures. The second axis is the hormonal axis, the endocrine axis, responsible for the second wave of the stress response. The target of my presentation is to disentangle possible biological mechanisms who play a role in the central fine-tuning of these limbs of the stress system. And because of its very rapid and short-lasting changes, we concentrated in the central autonomic network, the control center of the autonomic nervous system in the brain. So the objectives of my research is to explore biological factors that modulate central autonomic nervous system activity. However, we needed to somehow circumvent the cognitive input that majorly affects the autonomic reaction, so our thinking brain. That's why we deployed only objective stress challenges, either by pharmacological or endocrine substances. And in order to understand what's happening in the central part of the brain, we assessed the central autonomic activity by proxy, by heart rate variability analysis. So practically what we did is we gave a substance, we measured how it changed the heartbeat, and then we understood thereby what's happening in the brain. Our readout, heart rate variability, is probably one of the most established and non-invasive measures of autonomic activity and reactivity, and it's measured by the heart, an organ that's wonderfully and complexly innervated by both branches of the autonomic system. Heart rate variability gives us the variance between heartbeats, which is controlled by the two branches of the autonomic system, and high heart rate variability is a good thing, represents energy preservation state and high adaptability, while low heart rate variability is pretty much the opposite, and a very known mortality risk factor. Let's check out the 5 studies that are included in this thesis. I will start with the first 3 who employed the same stress paradigm. In the first 3 studies, we tried to understand the central role of the serotonin signaling system in the brain and the glutamate system in the brain on autonomic modulation. We did that by either providing drugs or assessing differences depending on genotypes of a transporter gene that has to do with serotonin signaling. We used the paradigm of a very known anxiogenic drug, CCK4, cholecystokinin tetrapeptide, which generates panic attacks in humans. We assessed baseline heart rate variability and then stress heart rate variability in order to see how this is affected by the serotonin and glutamate system. In the 2 first studies on serotonin, we found out that the SS genotype, so having the 2 short alleles of this serotonin transporter gene, is associated with lower heart rate variability at baseline, but also blunted autonomic reactivity after stress. And something similar we could also show in the second study that employed a long-term medication with a serotonergic drug that alters serotonin signaling in the brain. So people took 42 days of this serotonergic antidepressant, and we can also show that this led to a blunted autonomic reactivity. On the other hand, when modulating the glutamate system by a drug that actually enhances the glutamatergic signaling, we didn't show any baseline or autonomic reactivity differences in the 2 groups. However, we could show that enhanced glutamatergic signaling led to a better autonomic recovery after stress. The other 2 studies were endocrine studies. We used a different paradigm with hormones that either stimulate or inhibit the endocrine axis in order to find out more about the interaction between the autonomic nervous system and the endocrine system, and also how this is affected by history of stress in this paradigm depression history in our patients. The findings are very important because there are only few studies that actually directly assessed the interaction between the 2 axes of the stress system. We could show that endocrine axis stimulation led to reduced heart rate variability, which is a bad thing, And of course, a similar effect we could see in people with the same paradigm that had a history of chronic stress. So chronic stress led to lower heart rate variability after the stimulation. The interpretation of our findings calls for a very complex balance of the stress system reactivity at the central areas of the brain, a very overlapping one. Also shows a significant role of the central serotonin and glutamate neurotransmitter systems, a very significant interplay between autonomic and endocrine axis, and a significant effect of chronic stress history. In this next slide, which is very busy, I show you the overall comprehensive model with all the factors that are known to lead to alteration of stress reactivity. And with the small stars, you can see what we have tried to assess through our 5 studies, either with psychoneuropharmacology, psychoneuroendocrinology, clinical trials, but also genetic trials. Our studies used the objective stressors in order to circumvent the subjective cognitive input in the central part of the autonomic nervous system. The further identification of biological factors that modulate the individual stress response will be very helpful in the future, will help us with clarifying more about the etiology of stress-related disorders, where they're going to help us identify new biomarkers that will help us with individual risk assessment, the personalized prevention and progression and staging of diseases. What we need to do is to enhance research in this area in both genders and also different patient and age groups with more objective stress challenges, which have not been that easy to make. And we have to translate these findings into clinical findings in order for other medical disciplines to gain and understand more about their diseases. The technology will help us in the individual autonomic assessment and everyday assessment, but also with the identification of new treatment alternatives towards autonomic restoration. Taking home message, the brain-heart interaction will and can offer us better understanding of stress reactivity and its biological underpinnings, even though it's a highly complex area, where, however, balance is everything. And herewith, I would like to thank many people that helped me on my journey, from the Hamburg University, of course, the Maastricht University, and the VU University of Amsterdam and Athens. And I would like to close with my ideal homeostatic point. This is the place where I find inner balance, a small harbor in the island of Sifnos in Aegean Sea. And I would like to give back the word to you, highly esteemed Pro-Rector.
Thank you very much, Mr. Candidate, for your very nice talk, which is sometimes, at least for me, was a little bit challenging, as you call it. Some of the slides were pretty full, but again, we can talk about this later. And I would like to refer, since we have 1 or 2 minutes left over, that in your last slide, it's really tempting to be there and not to be here in our country. And I would expect that you would have closed with a phrase from a Greek writer and not with a phrase from Albert Camus. But saying this, we will continue. So we will start the opposition now, and the opposition will be opened by the chair of your assessment committee, which was Professor van den Hove, who is affiliated with our university in the field of neuroepigenetics, and I would like to give him the first word.
Thank you, dear Pro-Rector. First of all, dear candidate, congratulations with this very nice thesis, which was a pleasure to read. Also, an impressive track record with a nice list of publications. Also, my compliments to your promotion team and supervising it as well. Of course, some questions before we go to the fun part, although this can also be fun. I would particularly like to dive into chapters 2 and 3, which are sort of intertwined. Correct me if I'm wrong, but I think they were also part of the same sort of study, and they were that you made use of one of the subgroups and the other one, etc. Could you briefly comment on that, how they were aligned or dependent on each other?
Highly esteemed opponent, thank you for the question. The studies 2 and 3 are actually having a different design and different, these 30 volunteers that took place in the study. These were 2 independent studies.
Okay, so you were not making use of a control group of one for the other. Okay. Well, let's dive in, and that probably also explains some of the different findings you observed in the different studies. But in both the discussions of those chapters, you refer to the fact that you only assessed or you focus on people that were SS or LL carriers and not the heterozygous genotype. And you advise, well, it would still be interesting to do that. They were part of the actual recruitment, the heterozygous subjects. So could you still comment then maybe why you didn't assess them? You said, I focus on the extremes, which makes it more likely to find an effect. But I think if you include the ones in between, you might either find the dosing effect, or you could find that maybe having one allele of one of the 2 might be sufficient to observe a certain effect. So maybe could you comment on why you still did not assess those?
Yes, thank you very much for the question. Doing research in healthy volunteers and also with experimental panic provocation is very hard to do. That's why we also had very few people actually included in the studies. You're absolutely right. Having the intermediate genotype would be of high interest. However, there were practical reasons that led us to just include the 2 subgroups. It would be very difficult to recruit another 15 with the intermediate subtype, and also it was a study that was done in the clinical department, so it was something that we did on top of our job. So we had to look out, and being very honest, we had to look out for our time resources and money resources.
I expected this answer, and I know the drill, how difficult it is, so that's fine then. Similarly, in both chapters, but also here in the presentation, you explicitly mentioned that it would be of high interest to include females as well in your study. In your discussion, in the discussion of the 2 chapters, you refer to the potential confounding effects of contraception. I'm not sure whether that is a really strong argument, but you're probably going to tell me now that for pragmatic reasons you also did not include the females, or was it? I mean, because even if you can sort of try to control for contraception or take, is it then again for the same pragmatic reasons that you did not include them?
Well, there were some pragmatic reasons. Of course, menstruation is one thing that we know that influences stress responsiveness, but also we know from prior studies that males and females show a completely different profile in stress reactivity. So it would be a completely different data set. And of course, we would have to gather another 30, which takes us back to the pragmatic problems.
Yeah, and potentially more than 30, considering the confounding effects. Okay, now then let's zoom out and look again at those two chapters. Then in one of the 2 chapters, you did find a genotype times treatment interaction. And the other chapter you actually did not. And then you say, okay, for chapter 3, you then did a stratified analysis, and you see the same pattern, but it doesn't reach statistical significance. You mentioned that, but you don't explain why then you did not observe the same effect. Could you comment on that? What is your idea behind the fact that you could not sort of replicate that finding?
Well, this is a very nice question, and we struggle ourselves also with this question. We were happy to find a very similar effect, but not significant one. I can only guess that there were two differences in these two studies. The first study had just one one run-through, and the second study had two, so it was a crossover study, and the volunteers already had probably one run with the CCK4 infusion. So some of them had perhaps already the experience, and this might have influenced the autonomic effects. If I'm going through a situation that I already know, we know this is a factor that influences. The other one is that we had slightly different time frames between in the application of CCK4, and we know that circadian changes might influence. However, they were not that significant, so it was not that further away from the first study. So I'm guessing it's probably the first answer that might explain this missing significance.
Okay, good to know, and maybe combined with relatively low power. I mean, the sample size is not huge, of course. Okay, cool. My final question is on statement number 5. And actually, it's also related to some of this, well, you referred to that in your presentation as well. Maybe one of the panelists can read out statement number 5, which in the old and the new version of the statements is still number 5, I think.
Yes, stress research should focus more on objective stress challenges in order to avoid cognitive perception influence affecting stress reactivity.
Thank you very much. Also, based on your presentation, I think I understand what you mean. At the same time, stress is all about perception. So could you maybe explain in a bit more detail why you think we should focus more on this and sort of leave perception or exclude it from these type of analysis or work?
Yes, thank you. This is a wonderful question. Thank you so much for giving me the opportunity to comment on that. Of course, stress has to do with perception, and this is pretty much the problem that we are having as individuals with chronic stressors at the moment. Pretty much the majority of the stress paradigms have also the individual aspect, the cognitive aspect in them. Beginning with the trial-social stress test to also other ones. This has to do with personal memory, it has to do with personal perception, cognitive schemes with prior experience or not of the situation, with a lot of personal and individual traits of individuals. Applying objective stressors like pharmacological stressors is a way to see what's happening in the system without the influence or without the direct influence of the prefrontal cortex. Of course, everything that happens in our body will be analyzed again. If I'm having a panic attack through CCK4, my brain will notice it and will try to give its input. However, the first reaction will be without the filter, or in the most paradigms, let's say 80%, because there's always playing a role. And the objective paradigms are very hard to do, very hard to recruit volunteers to do that, but also to recruit patients to do that. And that's why they haven't been so numerous in the prior research. And I think this is one way to better understand fine-tuning and biological factors that play a role.
Yeah, so actually maybe one of the answers to one of the previous questions on the fact that chapter 2 and 3 differ, with maybe the repeated CCK in one of the two might actually prove your point, in the sense that perception in the repeated exposure might explain a different response.
Exactly.
And the same holds true then for the chapter on previous depression. Okay, well, I'm very pleased with your answers, and I give the word back to the program.
Thank you.
Thank you very much. The opposition now will be continued by Professor Klaas. He will be available online, so we have to look to the screens. He is affiliated in the Department of Depression and Stress Physiology at the University Psychiatric Center in Leuven in Belgium, and I would like to thank him that he has the time to join us this morning. And I'd like to give him the word, Professor Klaas, to you.
So thank you, Mr. Pro-Rector. Dear candidates, first I would also like, of course, to congratulate you on this impressive piece of work. I think it's really nice also that you try to look at the HP axis and the autonomic nervous system simultaneously and look at their interactions, and I think that's really important field. So congratulations to you and also to your promoter team. So some questions. Maybe also start with chapter 2, where you looked at the genotype of the 5-HTT-LPR, and you found that the SS carriers have a blunted reactivity to stress, at least looking at the HRV. In your introduction, you say that you expected an influence of this polymorphism on the autonomic response, which I think is okay. But did you expect this specific effect, or would you rather have expected that the SS carriers would have had an enhanced autonomous reactivity instead of a blunted one? So what was your expectation and why?
Highly esteemed opponent, thank you so much for this very important question, because we thought a lot about our results. And indeed, if we think back to the initial reactivity hypothesis, an enhanced reactivity would be probably expected by the SS carriers. The SS carriers have been related in several studies to worse cardiovascular events, but also higher risk for stress-related disorders, although this has been criticized in last studies. But our initial hypothesis was that the SS carriers will have the worst profile. So we were actually expecting higher enhanced reactivity. We saw reduced and blunted reactivity. However, we saw that together with a lower baseline heart rate variability before the stressor. And this leads us to the more modern studies that actually suggest that blunted reactivity is a major risk factor. To tell you the truth, after some years in this field, I'm not really sure we're going to find a definite answer, because stress reactivity has to do with so many different factors, and it could be a good thing to have enhanced reactivity at some point, and be also a good thing to have a blunted reactivity in others. I'm thinking about stress-related disorders with the two extremes, like depression with the typical and the atypical depression, or also PTSD with the classical and dissociative type, who actually show a mirroring of reactivity along the clinical case and clinical development. So it might not only have to do with the genotype, but also with the time that someone is affected by stress. Here in this study, we're now pretty sure having the worst baseline heart rate variability, that it's probably a bad sign to have blunted reactivity, as it goes together with lower vagal influence at baseline.
Okay. I thank you for this very nice answer, which also shows your profound knowledge of the field. I must say, in our experiments, when we do stress tasks in the lab, we find often the same thing, like a basal HRV which is decreased and a blunted reactivity after the stressor or in response to the stressor, which makes me think, but I'm not sure whether you would agree, that maybe when you have a long-term HRV lowering, so which means increased stress in some way, that maybe your stress system would be exhausted in some way and not able to react anymore to an acute stressor. Would that be a plausible hypothesis, you think, or how would you feel about that? There's a link between the basal changes and reactivity which is blunted in some way?
Thank you for the next question. Yes, of course. I think you're absolutely right. We have several things that's happening along the stress axis, along with years passing in stress exposure. There's a theory that the autonomic nervous system and the HPA axis diverge with several years of exposure, but also there's the theory of autonomic tiredness of the responses. Again, I think this has to do with time. We're dealing with a black box, I think, and we have to do several experiments to understand how all these biological factors affect our individuals. And I think time is one of those. And I totally agree with your hypothesis.
Yeah, thank you very much. I'm glad we agree. Yeah, that's nice. Maybe I'm going to jump to chapter 6 a little bit, where you looked at the effect of metyrapone on HRV in patients with a first depression and with recurrent depressions. And what I missed, well, two questions. What I missed a little bit there is a control group, a healthy control group. And I think you probably had those because I guess these are the people you described in chapter 5. So in chapter 6, why didn't you compare the two depression groups, first episode, recurrent, to healthy controls? Would that have been added value or not? Not really, you think?
Thank you so much for reading my dissertation so carefully. Indeed, we had the group in chapter 5. However, this group was not with respect to gender, with respect to age, smoking, etc. It was not really suited to do to use it as a control group. However, what is very interesting is that we see similar reactions in patients with prior depression as we see in healthy individuals in chapter 5. And what's very interesting is that patients with the first episode show a totally different stress reactivity. So this takes us back to the previous question of yours and my answer that time and exposure, long-term exposure to stress, as in repeated depressive episodes, might actually turn the thing around, and that's why we're seeing these differences. But to your question, we actually had to recruit another suit and another group of volunteers, which was again from the organizatory perspective pretty hard to do.
No, I quite understand. I was a bit worried, of course, when I saw in this study that your first episode MDD group only contained 5 people. So if I remember well, so which is a very small group. So were you worried about the power or worried about how to interpret these results given the small sample size you had there?
We worried about the power. And again, I found it even more difficult to recruit patients than volunteers. It was a very hard job. We used 2 years in order to recruit those 14 patients that were willing to do this study with us. We were, on the other hand, happy to see those significant results, even in the small group. However, of course, we have to reassure and do another study with a larger N on this aspect.
No, I fully understand. Problems of recruiting patients, and especially depressive patients without medication, is very, very difficult. So I fully appreciate the efforts. And with that, I would like to thank you very much for this discussion, and I would like to give the floor back to the pro-rector.
Thank you very much. The opposition will now be continued by Professor Linden. He is affiliated and appointed as professor in translation neuroscience at our university, and I'd like to give him the word.
Dear candidate, thanks very much for the very clear presentation. We learned a lot. It was complex diagrams of neuroanatomy and circuits. I'll come back to this in a minute. I'd like to congratulate you and your promotion team. Very impressive international collaboration. I was very pleased to be part of this, Corona, and to ask you one question about Chapter 3 in relation to the introduction, because in the introduction you list various components of the central stress system. This is on page 10. You probably don't read reminding, but this is where the different components are listed. And which of these did you manipulate with the serotonergic manipulation, the escitalopram treatment in Chapter 3?
Highly esteemed opponent, thank you so much for this question, which is indeed a very hard question to answer, because we actually don't know. When it comes to the serotonergic system, we have 14 different receptors along the brain, and of course, giving a patient a serotonergic drug that is not that sensitive and goes to all receptors could affect the serotonergic system in several parts of the brain. However, our understanding goes to rather structures who have a very high density of serotonergic receptors, like the amygdala, for example, or Raphe nuclei in the pons. And we think that these might be the most core structures that are influenced by the serotonergic medication. However, as I stated before, we're not sure, and we cannot really prove that this is the case.
Now, you elected to manipulate the serotonergic system to address the central, I think, you think mainly central action here in the manipulation. You could also have manipulated this induced panic response by dual peripheral and central, or mainly peripheral action, perhaps with adrenergic or anti-adrenergic or cholinergic medication. Is that also something that's being done with this pharmacological challenge, and what's the literature there?
Well, thank you for the question. Indeed, there have been some studies done by our research group, also using other substances like the ones you mentioned. However, I'm not really sure that I'm pretty sure we didn't use the CCK4 paradigm in all of them, but in some of them. So it was a collaboration done with Professor Lars Schwabe from Hamburg, and he did some experiments there. However, I was there not the PI, so I can only comment very briefly on those studies.
And this is coming back to the proposition 5 that was already read in terms of the subjective, objective measurements, and perhaps the sort of classical models of stress, or more generally emotion responses and embodied emotion that are coming back now. How would it feel for someone just to have a peripheral attenuation of a panic response?
Well, thank you for this nice question, because it gives me also the opportunity to comment on the overlap of these brain centers that are responsible not only for autonomic activity and reactivity, but also for emotional regulation and also cognitive regulation. Most of the parts of the brain in this central autonomic network have overlapping influence on all these three major parts of the human experience. How does it feel to have a panic attack? Well, it's something that's probably the other way around, if we compare it with a classical panic attack, which starts with a cognition and then goes over to the panic, the somatic response of the panic. Here we probably have quite the opposite, which sometimes also happens in panic patients. They're having the sensations, and then they turn on the cognitive part, which evaluates what's happening. So getting back to the question of the former opponent, that's why the first time receiving a CCK4 is probably also the most objective one, and the second time probably not.
Thank you. I am aware of limited time left, so I'm not going to pursue this very interesting discussion, but I didn't want to miss the opportunity to refer to one of the Greek authors you mentioned, because Pro-Rector reminded us that we really needed to discuss the Greek literature a little bit as well. You cite in Proposition 9 Hippocrates, and perhaps your panel can read the English translation of that proposition for us.
It is not possible to know medicine without knowing what a human is.
Thank you. So how does this statement relate to your thesis?
Thank you so much for giving me the opportunity to comment on Hippocrates. Well, I'm feeling very humbled to do that. However, my understanding, and not just from my thesis, but also my clinical work, is that every individual is different. The reactivity, the stress reactivity of every individual is different, and is influenced by so many different objective and subjective factors. In order to be able to help people, we have to really understand how they function, and we can only do that by observing symptoms, observing behavior, observing cognitive patterns, and also emotions. We can have two very similar patients with similar symptoms, but they can vary so much that we have to pursue totally different treatment aspects in order to help them. So observation of the individual emotion, reactivity, and cognition is for me the key to understand the people and help the people.
Thank you very much. And I hand the word back to the Pro-Rector.
Thank you very much. Then the opposition will be continued by Professor Blokland. He is appointed as Professor in Comparative Neuroscience at the Faculty of Psychology and Neuroscience at our university. And I also like to give him the word.
Thank you, Pro-Rector. Yeah, actually, some of my questions that I tried to prepare for you now already taken away from my colleagues. Thank you very much, colleagues. Anyways, I still have some questions, but it will boil down to the things that maybe already have been said, and maybe I'm not really convinced yet. Anyways, but first of all, I would like to, of course, congratulate you on your wonderful thesis and your wonderful work that is described there, and of course your supervisory team who helped you to compile this really nice thesis on probably a part of your whole repertoire that you already managed to do. Yeah, so we were talking about statement 5. We don't have to read it again. And I would like to come back maybe to Hippocrates again. So when I would like to go to page 14, where you have this you like complex schemes. That relates, I think, to the complexity of man. And so when you have the stressor, you see that it's all. So in this model, the stressor is going to the cognitive box, and then it goes all the way down to the central nervous system and the peripheral nervous system. Yeah. So I'm missing actually some arrows that go backwards. Why did you're nodding now? But why didn't you do it?
Highly esteemed opponent, thank you so much for pointing this out. Of course, there are arrows. There should be arrows pointing up. However, you already mentioned how highly difficult it is to have a simple presentation, and I think it was a little bit too much to do the backwards arrows as well. Of course, there are many signals from the body going to the central centers of the autonomic nervous system and also the hypothalamic area, from ventilation to CO2 in blood to baroreflex to you name it. There are so many. We're still trying to disentangle where they actually work on, what they influence, because we know they highly influence autonomic reactivity as well. And it would simply be just too complicated to have all the arrows in there.
Okay, yeah. All right, well, more work to do, of course, and as we all have to do that. Okay, having cleared that, and we agree that this is maybe a too simplistic view. In your thesis, I think the main thing is that you would like to relate this autonomic nervous system with something like depression or anxiety, panic. But when I read on page, 45, where you report the results of study 2, I don't read, I read that there is no correlation between the reactivity of the autonomic nervous system as measured by heart rate functions, heart rate, and the API scores. So to what extent do you think that the autonomic nervous system reactions relate to the more psychiatric conditions? Because I don't see the correlation. Did you make scatter plots to have a look maybe if there are some outliers? Because you have only small samples, one outlier could already maybe disturb this whole correlation. But did you have a look at that? And then the second question is, why don't we see this correlation?
Thank you so much for this interesting question. Indeed, we have looked at this. Of course, there were some outliers as always. However, this is something that we actually know pretty well from research. We have done panic provocation in my lab for a long time, and there were only probably half of the studies that we've done that we actually saw a correlation between the physical response and the emotional response. We cannot really understand why this is the way it is. Most of the paradigms were done through CCK4 challenge. So it was the same challenge, the same milligrams. However, we cannot really understand why this was. And this has been also shown in some later on studies by our group. Some of them found an association and some of them didn't.
Mm-hmm. So that means that there is a correlation then between, do you think there's between these autonomic nervous system variables and the psychiatric conditions, or not?
Well, it might be, but it might be on an individual level, since some volunteers did not show the same emotional response to the stressor. It goes back to the individuality of each and every one of us.
So that means that statement 5 is maybe a little bit overestimated, that because you talk about an objective stressor, when now you see probably it's much more complex and arrows going back to the cognitive perception than having its effect again. Yeah.
Definitely, yes, yeah. I agree.
Anyways, but although it's very complex and cognition is always in there, do you think that if you would is a treatment with drugs that affect the autonomic nervous system would have a beneficial effect in psychiatric conditions? Like depression like depression, for example. If you would treat people with a drug that affects the autonomic nervous system in a certain way, that you could relieve some symptoms because you suggest that they are related to each other.
Okay, thank you for your question. And of course, this is something that also very complex to answer. But what we know is in non-patients that these drugs have some effects. Now, in patients, there are several studies that show some kind of normalization in some patients. Again, not in every patient. We have normalization of autonomic responses with respect to remission or partial remission. It's also associated to the chronicity of the disorder. Some people having the disorder longer don't really show normalization of autonomic measures. However, what we do know is in a clinical perspective, patients with depression receiving antidepressant medication like the SSRIs show a reduced risk for cardiovascular disease or relapse. This is something that we do know from broad epidemiological studies, so there must be something there.
Yeah, but probably it's more complex. Yeah. Yeah, but I also wanted to go to statement 11, your proposition, because I think Hippocrates was quite vague, because he is talking about medicine for humans. But what do you think he meant with human?
Well, Hippocrates probably meant the idea of a human as a thinking creature. This is probably answered before in my answer to the previous opponent. The thinking is what brings us to feeling and reacting. And to the ancient Greeks, a person without thinking wasn't a person. So I probably think he's targeting the thinking component, the cognitive component of our nature.
Yeah. Because I think that in medicine, as we know it now, some drugs work without knowing. An antibacterial drug. So, but if we go to psychiatry, then it becomes more complex. That although we try to treat biology, we don't not always treat the psycho.
Well, I totally agree with you, because in psychiatry we have to do, again, with overlapping systems. We know that an antiserotonergic antidepressant works the same as 1 hour of walking every day. We know that psychotherapy alters biological functioning, and we know that it alters also interconnection between brain areas. And we know that it's a holistic approach that we need in order to help our patients. Yes, psychiatry is very complex, as my diagrams today, and that's the challenge. That's why we're the best discipline.
Okay. Yeah. I would like to give the word back. I would like to tell you something about what causes what, the biology of the psyche, but that's another question. Maybe we can talk about that later. But I will give the word back to the prorector.
Thank you very much. Now the opposition will be continued online. We go to Dr. Kwaadvlieg. She is affiliated with the Department of Neuropsychology and Psychopharmacology at the Faculty of Psychology and Neuroscience, and I would like to give the opposition the possibility to discuss with you. Dr. Kwaadvlieg. And we noticed you have corona, and we saw the evidence right now.
Thank you, Pro-Rector. Dear candidate, can you hear me well?
Yes, we hear you well.
Yeah, okay, because I do hear myself as well. Okay, good. I also have read your thesis with a lot of interest. I learned a lot. I think it's a nice, coherent set of experiments. So in these congratulations, I would also like to involve your promotion team. And I think also this collection of endeavors adds yet another layer to your already, in my view, also impressive toolkit. With your clinical experience really standing promptly in there as well. So you have a diverse accomplishments, which I would like you to congratulate you with as well. I found myself captivated by the idea expressed actually on page 15, namely that stress systems reactivity to stressors plays a crucial role for adaptive responses, survival, and overall health. And you support this idea, it's illustrated on page 16 in figure 3. And this is the figure that shows like 2 lines. One, the solid line, which shows the adaptive response, and one is the dotted line showing the disrupted reactivity, which in your view is then really important for mental health. Upon closer examination of this figure, however, I would like to propose an alternative interpretation. What I see is not a disrupted reactivity characterized by this upwards ascent until the peak, but rather a disruptive recovery. And this would be exemplified by a downwards trajectory after the peak. So if I'm looking to the figure, I see that the 2 lines start to deviate from the second peak onwards, so the second stressor. And again, in my view, the difference is in the downregulation after this second stressor, which is like in the dotted line seen that it's not that much, that it's insufficient. So do you share my view that the emphasis in research, but also actually maybe in your thesis, should shift from disrupted reactivity to disrupted recovery? I look forward to hear your perspective on this.
Esteemed opponent, thank you so much for this comment and your question. It's really hard to answer that, if it's reactivity or recovery. Well, we have several studies that actually suggest that recovery from our stressor gives a better understanding about where exactly the balance is at the moment in the living organism. However, of course, the reactivity is very important as well, especially also in psychiatric disorders like PTSD. So it's very hard to answer which one of the two is more important, and some of my results also back this statement. For example, in the 3rd study on glutamatergic signaling, we don't find an effect on baseline heart rate variability or reactivity to the stressor, but we do find an enhanced recovery from the stressor half an hour later. So I think this is pretty important as well, and it might have to do with different signaling systems in different brain areas, because they're not the same areas who take control in the first part and the second part of the stress reaction, and also the recovery. So as much as I would like to totally agree with you, I'm not really sure if I could pick one over the other one.
Okay, good. But if we come back to the figure, what do you think of my interpretation that the figure shows? Because your interpretation is that it's reactivity.
I'm not really sure. Could you perhaps repeat the question? I couldn't understand.
I think there is a delay. I will talk slower. So I was talking specifically about figure 3, which you would interpret like disrupted stress reactivity. What would you say about my interpretation about disrupted recovery?
Okay, about figure 3. So now I got you. Thank you so much. Well, it is a very simple figure. It's not a figure that shows us what's actually happening. Of course, here recovery and reactivity should have the balance, and this figure gives a little bit more credit to one of those. What I can promise is that I will work on a new edition of the figure, and hopefully we can jointly publish it somewhere.
That sounds as a promising idea. So you also said like it's really different to differentiate recovery, reactivity. So if we would design then also a full new study to investigate it, and maybe we do want to use again a pharmacological challenge, because your view is that we should stay on an objective level and try to exclude the psychological mechanisms in stress. Would you have an idea of a pharmacological challenge that we could use?
Thank you for your question. Just to be perfectly honest, I was not suggesting just using objective challenges, but using more objective challenges in research, which is pretty hard to do. Well, objective challenges can be a lot, like CO2 inhalation, which is done here in Maastricht by my supervisors, and indeed there are some several publications on this one. So CO2 inhalation, then there are several other panicogens like caffeine infusions, but also the ones mentioned before, neurogenetic drugs, but also other endocrine challenges. They are a lot, and we can also use also perhaps neurostimulation.
Dear Mr. Candidate, you are allowed to finish your answer, but you're not obliged to. So whatever you want.
Thank you so much. I think I have just finished. Thank you.
Okay, good. You were just finished. Dear Mr. Candidate, dear Mr. Agorastos, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss the quality of your thesis, but particularly the way you have defended your thesis this morning. And I would like to await you and your partners our conclusions and our return to this office. Thank you.
Thank you so much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting the west side. No place like home. If it's questions that you've got, go the extra mile or die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Mr. Candidate, dear Mr. Agorastos Agorastos, double name, the degree committee here present has assessed not only the quality of your thesis, but in particular the way you have defended your thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Schuurs is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take the floor. Professor Schuurs.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and conformity with the decision of the committee here present, I hereby confer upon you, Agorastos Agorastos, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
Sehr geehrter Herr Dr. Agorastos, lieber Agorastos, allererst herzlichen Glückwunsch zum Doktortitel, auch an deine Familie, deine Frau, deinen Kindern und ja, deinen Eltern natürlich. Unsere Wege haben sich zum ersten Mal in Florenz beim Sommerkurs des damaligen Master in Effective Neuroscience gekreuzt. Bei unserem ersten Gespräch im Garten der Villa Fenali war sofort deutlich, dass du intelligent und neugierig bist. Im positiven Sinne natürlich. Aber wie intelligent und neugierig durfte ich dann in den Jahren danach erfahren. Deine Doktorandenzeit hast du zum Teil in Hamburg verbracht, in Deutschland. Ein Grieche, der in Wien seine medizinische Ausbildung gemacht hat und dann seinen Weg nach Hamburg gefunden hat. Deine Kollegen denken noch sehr gerne an deine Bewerbung zurück. In deiner Bewerbungsmappe lag ein riesiges Foto von dir. Du hattest einen weißen Anzug an, lange gewellte Haare und einen Rauschebart. Schnell freuten sich deine Kollegen auf leidenschaftliche Diskussionen über die Ägäischen Inseln und Zypern. Einmal in Hamburg angekommen, hast du deine Umgebung nicht nur mit deinem griechischen Charme beeindruckt, sondern auch mit den Studien, die du durchgeführt hast. Du hast verschiedene Methoden gelernt, kritisch hinterfragt und angewendet. Von genetischen Analysen bis zur Stressprovokation, wie wir heute gehört haben. Und während du im Labor Probanden gestresst hast, hat das Leben dich auf deine Stressbeständigkeit getestet. Wir wissen alle, dass die Arbeit als Wissenschaftler manchmal sehr stressig sein kann. Literatur lesen, Experimente durchführen, Daten auswerten und am Ende auch noch aufschreiben. Und ganz zu schweigen von den vielen Kongressen, die du auch noch besucht hast. Neben der wissenschaftlichen Arbeit hast du auch noch als Arzt gearbeitet. Patienten sehen, wichtige Entscheidungen treffen, Nachtdienste haben. Alles sehr anspruchsvoll. Nicht zu vergessen, zuverlässig und hilfsbereit zu sein. Und all diese Aufgaben hast du mit Bravour gemeistert. Deine Kollegen sprechen noch heute in den höchsten Tönen von dir. Du warst selbst noch so weit unter deiner maximalen Stresstoleranzgrenze, dass du schlaflose Nächte durch 2 wundervolle Kinder obendrauf genommen hast. Wenn man sieht, wie beeindruckend du deine Aufgaben gemeistert hast, dann fragt man sich doch, was dein Erfolgsgeheimnis ist. Die Umgebung, die du vorhin in deiner Präsentation gezeigt hast, die hat sicherlich mitgeholfen. Und natürlich hast du auch von Anfang an wichtige Eigenschaften wie Intelligenz und Motivation mitgebracht. Aber die eigentliche Eigenschaft, die hinter deinem Erfolg steht, ist doch vielleicht eine andere, denke ich. Vor einigen Jahren hast du dir in Hamburg ein Zimmer mit einer Psychologin geteilt. Kurz zurückdenken. Eine deiner Aufgaben als Psychiater war es, Patienten zu sehen und ihnen bestmöglichst zu helfen. Und viele Patientinnen tuschelten im Wartezimmer, dass sie gerne von dir behandelt werden wollten, weil du immer so tolle Ratschläge geben würdest. Nach einem gemeinsamen Gespräch mit der Patientin wartete die Psychologin also gespannt auf deinen Rat. Was wird er der Patientin sagen? Was für tolle Ratschläge gibt der Patienten? Du hast dich leicht im Stuhl zurückgelegt und dann mit aller Überzeugung gesagt, heute machen Sie mal was Schönes. Essen Sie was Gutes. Für eine Patientin und eine Psychologin ist das vielleicht nicht gerade der Ratschlag, den man von einem Psychiater erwartet. Allerdings stellte sich heraus, dass dies genau die Intervention war, die die Patientin in dem Moment brauchte. Ja, deine Überzeugungskraft hat bestimmt auch geholfen, aber auf jeden Fall, sie nahm den Ratschlag an und war dir dankbar. Ich denke aber, dass du diesen Rat nicht ohne Grund gegeben hast, sondern weil du selbst ein echter Lebensgenießer bist. Und dazu gehört nun mal gutes Essen. Deine Kollegen schätzen an dir, dass du ein ausgezeichneter Partner bist, um gut essen zu gehen und auch um legendärische Partys zu feiern. Deine Lebenseinstellung ging sogar so weit, dass du als einziger Mitarbeiter an der Abteilung in Hamburg eine Espressomaschine und einen Mini-Kühlschrank hattest. Sogar in der Mensa warst du bekannt, weil du als einziger Mitarbeiter Eis in deinen Espresso haben wolltest, um Kaffee Fredo trinken zu können. Vielleicht ist diese Eigenschaft von dir, nämlich dass du ein echter Lebensgenießer bist, das eigentliche Geheimnis deines Erfolges. Ein Lebensgenießer zu sein, hat dazu beigetragen, denke ich, dass du den Stress besser hinter dir lassen konntest, um dein inneres Gleichgewicht zu bewahren. Der Titel deiner Dissertation, The Balance Within, und die verschiedenen Elemente, die aufeinander gestapelt sind nicht nur ein Spiegel deiner Studien, sondern eigentlich auch von dir. Lieber Agorastos, ich hoffe, dass du auch in der Zukunft dein Gleichgewicht bewahren kannst und vielleicht noch die ein oder andere Kugel extra balancieren kannst. Ich wünsche dir und deiner Familie alles Gute für alles, was vor euch liegt. Herzlichen Glückwunsch. I'm giving the word back.
Also lieber Dr. Agorastos, es wäre schon selbstverständlich attraktiv zu sein, um weiter in Deutsch zu reden, aber ich werde doch schnell in Englisch. Ich hoffe, das ist auch genügend. I would like to also congratulate you on behalf of Maastricht University about our Board of Deans with this new title, which you was looking for. And I would like to also put in my congratulations your promotion team, who brought you here, and I checked here all the files, which was very impressive. I also would like to send my congratulations to your family online somewhere in the cloud. I noticed they are somewhere, I'm not sure, in Germany, I suppose. And I would like to also congratulate them as well. I also would like to thank the members of the Corona, and particularly the two online, Professor Klaas and Dr. Kwadwijk. Thank you for joining this morning and that you took the time to stay online the whole period. I also like to thank the members here who came this morning here to this meeting. Just a personal note, I was a little bit intrigued by your last slide, and I made a little quote on here, and I thought, what is he meaning here with your take-home message? So basically, if I quote correctly, and you don't have to answer anything, so don't worry about it. You mentioned here, balanced stress is basically good. Is that correct? Yeah. And I wonder, how do you feel this morning? Was a thesis defense for a thesis candidate a balanced stress? Because the way you defended to me was that you enjoyed it. So basically, this stress was a good stress for you. And by that, I'm closing here, and we're going to do some coffee and pie, most likely, and make some pictures. I used my hammer and officially close this session. Thank you very much.
Please come forward, Dr. Agorastos. Panelists, please join. Supervisors, please join. You stand here. Recording stopped.